is much better. At least I hope it is. Then I just realized that I can see chat on my phone. Beautiful. Sir? Oh, it's probably... Oh, it would have been nice if I could chat from here instead. I keep messing with my hair because this is making me very nervous. But anyway, okay. I'm just gonna let it flow and see how it goes, and if it's another failed attempt, this is my third attempt, then I'm gonna cross out OBS from my stream and just stick to streaming on YouTube. Alright, so let's get back into it. I left off on braided behavior, and I'm gonna keep doing that for the rest of the night. And then, yeah, let's just get into it. I hope you can hear me well. If you can't hear me well, I changed the settings for the audio, so that might be why. Hopefully not. It says you can hear me. Let's go. Okay, yeah, we left off with the transmitter release, exocytosis. I didn't try opening this last time, so hopefully things are okay. Start going over this in discussion. Samples, responsiveness. 28 dropped frames. Oh my gosh. CPU is really getting up there. Yeah, so I'm watching some of the stats here at the bottom. Because I'm dropping a lot of frames. Stream quality is good. Fascinating. I don't know, man. Oh, this is what I needed. Okay. Amino acids contain the main group H2, neuropeptides, acetylcholine, monoamines, purines, gases. I don't know any of these. Well, I recognize some of the terms, but I don't know any of them. I haven't been pulling my day on it. Purines, gases. Okay. What the? What? Interesting. Okay. have my mic quite lowered. What did I have it at? Is that better? OMG. What? Is that better? The sound coming from me? I did lower it a lot because I felt like I was too loud. I'll raise it more, but I think that might be too loud. Okay, we're gonna keep it there. Okay, I'll keep it there. Let me know. Okay, so amino acids. Yeah, because I talk a lot, so we'll see. Amino acids. Acids containing an amine group. Amino acids. Acids containing an amine group. NH2 amine group. That's nitrogen, hydrogen, and that's one nitrogen and two hydrogen. An amine group. Fascinating. Chain of amino acids. So that's multiple NH2s. Chain. Neuropeptides. Amino group acids. Neuropeptides. Amino acids, neuropeptides. The names are so different. Neuropeptide. I know peptides are like peptide bonds. Neuropeptides, bonds, neurobonds, amino acid bonds. I don't know. I really want to look them up. I think what I'll do. Okay. So I disconnected from Wi-Fi on my phone, so I'm going to use my phone as my search engine because I'm trying to keep this live alive. Short chain polypeptides, poly meaning multiple peptides. Peptide. Any of the group compounds which act as neurotransmitters. Group compounds act as neurotransmitters. Okay. At a synapse, one neuron releases chemicals that affect a second neuron. These chemicals are known as neurotransmitters. Chemical that affects a second. Thank you. It literally says, do not confuse nitric oxide, NO, with nitrous oxide, N2O, sometimes known as laughing gas. Thank you. Because I was literally thinking that. I hope I'm not still frozen. I shouldn't be. I'm scared. Because I'm having a really tough time understanding these, like making associations in my brain and making them make sense. Neurotransmitters, amino acids, neuropeptides. I haven't even gotten to the other 4. Amino acids, neuropeptides, amino acids. Yes, acetylcholine. Chemical similar to an amino acid, except for changes. Yeah, that's a carbon hydrogen 3 group. A 1-member family. That doesn't make any sense, but okay. Monoamines. Neurotransmitters containing 1 amine group. Okay, things are starting to make connections. Of them. So we have the monoamines. These are all neurotransmitters. Transmitter with 1 amine group, NH2 group. Amine group, NH2 with 1. Formed by a metabolic change in certain amino acids. Okay. Amino acids are acids containing 1 amine group. Okay, so 2 different things. Monoamines, amino acid. It's a monoamine with an acid. Oh my god, okay, that makes sense. Okay. Monoamine, amino acids. It's an acid containing a monoamine, an amine group, NH2. Got it. Neuropeptide is a chain of amino acids. A chain of acids with an NH2 group. Okay. Acetylcholine. Chemical similar to an amino acid. So it's an acid, but with an NCH33 group. It's wild. So acetylcholine is like its own thing. It's an acid with an NCH33 group. That's wild. Okay, the two separate ones that are like different from the four are purines, category of chemicals, including adenosine, and several of its derivatives, and then gases include nitric oxide and possibly others. Nitric oxide being the most important because it has to do with controlling blood flow to certain areas of the brain. Okay, cool. Now we're gonna get into the neurotransmitters. Energy. I'm gonna start with monoamines because that makes more sense to me. Monoamines. Instead of also modified from. Okay, sure, but it makes sense the other way for me. So types, we're going to do indoleamines, which includes serotonin. Interesting. We have catecholamines, dopamine, and norepinephrine. Oh wait, no, that's a catecholamine. Dopamine with an E at the end. Epinephrine. And we're going to learn what those are. I kind of have an idea of what some of them are, but not all of them. Okay, so those are monoamines. Fascinating. Okay. Catecholamines. That scared me. Bold italics. Yes. Okay, good. And then we're going to go with amino acids. This include glutamate, GABA, glycine, aspartate. I've heard of aspartate before. Maybe others. And then after amino acids, we have peptides, neuropeptides, which I guess they're just calling peptides. Chains of V, I owe you food. Let me know what you want for food. Endorphins. Fascinating. Substance P and neuropeptide. Oh, neuropeptide Y. also has the peptides. Pink. Purines, gases. We have ATP for purines. Or here's the category of chemicals. Adenosine. Maybe others. Yeah, because we haven't discovered all of the neurotransmitters in the body. There's like over, what did it say, 500 of them? We've discovered over 500 of them. Something like that. I don't remember where I read it, but yeah. Gases. Yeah, nitric oxide, nitrous oxide, maybe others. Okay, let's try to memorize some of these. I finally understand what amino acids are, monoamines, neuropeptides, acetylcholine, apparently it's its own thing, purines and gases. Right, okay, so monoamines, what do they include? We have endoleamines and catecholamines. How do I get to remember those? Endoleamines, which equals serotonin. That is a monoamine, serotonin. That I remember. Then we have catecholamine. Okay, that one has 3 monoamines. These are like the main ones that we hear almost all the time. Dopamine, norepinephrine,
it's norepinephrine. Norepinephrine. Dopamine. No. Oh yeah, dopamine. Norepinephrine. Norepinephrine. And then epinephrine on its own. Those three for catecholamines. Monoamines. Mono means being just one NH2 group. NH3, NH2, right? N and 2 hydrogen, NH2. Okay. All right. So far we have monoamines down. Monoamines we have indoleamines. I already forgot. Indoleamines, right? Serotonin, it's monoamine. And then we have catecholamine, which includes dopamine, norepinephrine, epinephrine. Those three monoamines, right? Then we have a modified amino acid, acetylcholine, which is an acid but with an NCH3 group. It's a nitrogen with attached to a carbon, attached to 3 hydrogens, and then there's 3 of them attached to 1, just wild. Yeah, see, like it explains it here. That's an NH2, carbon, HOOH, catechol. Yes. Actually, let me look that up. And let me find acetylcholine chemical. Oh, it's right there. Okay. Yes. Maybe. So I was right. It's a nitrogen attached to the carbon that then has 3 hydrogen bonds. And then there's 3 of them. I was right. Amine group, which is but then here it's showing that it's bonded to another oxygen. It has double bonds on oxygen and another CH3 on this side. What? Acetic acid and choline. Let me look at acetic acid. So it's the acetic acid. Of course, carbon doesn't this one's carbon. 3Hs. Okay. And then choline. Choline is that one section. The N plus MOH. So that part is choline. And then this is the acetic. But it's acetylcholine together because of that. Because this O has, so it's carbon. I'm not sure why, but it makes sense. But that's how I'm gonna memorize it. So acetylcholine, it's made up of 3 CH3s surrounding one nitrogen group. So it's not an NH2, it is NCH3. Right, okay. And that's just acetylcholine. It's a modified amino acid, so it's a subcategory of an amino acid. Now we're getting to the good stuff. Amino acids. We have glutamate, I don't know what that is, GABA, glycine, aspartate, and maybe others. Those are the ones that they want us to learn. I want to learn what they are though. It helps me memorize stuff better if I know what they do. We'll probably, I have a feeling we're going to learn what each one of them do later on. So I'll just try to memorize them. So we have 4 amino acids. For monoamines, we also have the 4. Dopamine, serotonin, norepinephrine, epinephrine. For amino acids, we also have 4. That's cool. Glutamate, GABA, glycine, aspartate. Glutamate, GABA, glutamate, GABA, glycine. Glutamate, GABA, glycine, aspartate. So 3 Gs. Okay. Glutamate, GABA, glycine, aspartate, and the others. Glutamate, GABA, aspartate, glycine. Okay. So those are types of neurotransmitters. Okay. Then we have peptides, chains of the amino acids. Okay. So there's 3 of those. We have endorphins, substance P, neuropeptide Y. Why they're called that, I have no idea. So neuropeptides are just peptides. Peptides, we have neuropeptides. Neuropeptide Y. Neuropeptide Y, whatever this was, P, and endorphins. Substance P, whatever this was, P, substance P. Makes sense. Neuropeptide Y. And that's perfect because I have no idea why peptide Y. My memory is hilarious. Peptides. Neuropeptide Y. Okay. And then we have substance P. Endorphins. So like we've already heard of some really popular ones. Serotonin, dopamine, epinephrine, norepinephrine. I already forgot the other ones. I already said serotonin. Acetylcholine. Yes. Okay. So those peptides, chains of amino acids. So endorphins have chains of amino acids. So they're not very simple. These are a lot bigger. Neuropeptides are bigger. Amino acids being the tiniest, but also not the tiniest. Amino acids, do you know what those? Glutamate, GABA, glycine, aspartate, and the others. And then we finally get to purines, which are chemicals. We have ATP. I've heard of ATP before, but I can't remember what it was. Adenosine, and other stuff. So those are the 2 main ones. Okay. So monoamines, we had the 4. Amino acids, we also have the 4. And then for acetylcholine is just this modified one. That's fine. And then we have the peptides, which is also 3. So it's 3. So 4, 4, 3. And then for purines, we have ADP and adenosine, so that's 2. And then for gases, we have 1 as well. So modified amino. Gaseous being nitric oxide. Okay. So I was just trying to remember what those are. Now I think I'm doing pretty good. Alright. So the neuropeptides and NO serve special functions, right? Neuropeptides being glutamate, GABA, glycine, aspartate. No, there's only 3 of them. Neuropeptides being neuropeptide Y, brain work, substance P, and endorphins. It has served special functions. Okay. All but a few transmitters are amino acids, derivatives of amino acids, or chains of amino acids, which is the big 4. The most surprising exception is nitric oxide, a gas released by many small little neurons. Fascinating. Okay. Nitrous oxide is N2O. Okay, nitric oxide is poisonous in large quantities and difficult to make in a laboratory. Fascinating. Yet many neurons contain an enzyme that enables them to make it efficiently. One special function is nitric oxide relates to blood flow. When a brain area becomes highly active, blood flow to that area increases because the gas is released. How does it become more active? Message comes from the gas released, nitric oxide. Many neurons release when they are stimulated. Inhibition opens similar to nitric oxide dilates, opens the nearby blood vessels, thereby increasing blood flow to that area of the brain. Fascinating. Okay, nitric oxide, what do I remember? When the neurons release NO, vessels dilate, which increases blood flow to that area. It's hard to make in a lab, but the neurons make it very easily. How is it doing? It seems to be doing well, so that's cool. Oh, hi Ruben Tan. Sorry, I don't check the chat too often, but I try. Hi Ruben. I also don't hear any alerts, so I'm sorry. Okay, so that's nitric oxide. Okay, when neurons release NO, blood vessels dilate, which increases blood flow to that area. What else does it do? That's basically it. synthesis of transmitters. I don't even think we started talking about that. Start talking about neurotransmitter uptake. Okay, so that's that. It's basically this process where we have the presynaptic terminal, and then in that terminal, there's an influx of calcium when an action potential occurs, because there's not much calcium inside, all that's outside. Kind of like how when I totally just learned this. What is it called? Potassium, and it wasn't chlorine. Sodium. Sodium. Okay. Basically, like an action potential when. Thank you. So I got distracted. Sodium is low inside the neuron, so that was the influx which causes the action potential. You reach that threshold. Yes, okay. Same thing at the presynaptic terminal. We have vesicles which carry the calcium ions and neurotransmitters. The calcium ions allow the vesicles to fuse with the membrane, which allows them to leave, right, into the synapse cleft. What is it called? It's something cleft. I think it's synaptic cleft. Synaptic cleft. I've lost it. So this is the vesicle fusion, right? So this is what I'm talking about. But this is like the real life one, not the really cute image. Fancy show. Yes. Neuropeptide transmitters, dense cord vesicles, compound. And so if you're talking about that. MT transmitter is cleared from the synapses by two active processes: uptake by transmitter molecules, mostly into glial cells. So yeah, that's after they exit, then they can get back into, it's this one image, this one. I'm gonna screenshot this. It's gonna be, what do I want to call this? Oh, wait, I put it in memory. This is the important one. So once they are released through the membrane, some of the vesicles come back. Neurotransmitter uptake, reuptake. Okay. So transport molecules transport protein. This uptake mostly into glial cells. Yeah. Transport protein. Food uptake. Secretion of receptors. Transfer to release. Post-uptake cell releases retrograde transmitters that slow further release from presynaptic cell. The glial cells neurotransmitter transports these balloons, these vesicle recycling, endocytosis. Yeah. Okay. Vesicle recycling. So there's two processes going on here. Transports food from the synapses back to the Yep. Okay. Now we're getting into inotropic and metabotropic receptors. We were learning about these in discussion. It was going so fast I didn't care. It's transport and storage of transmitters. I think I should read this. Release and division of receptors. I think he shows this process here. Inactivation and reuptake. Okay, so this is where we're at. Oh, it's a acetyl group. Huh. I'm not the best at chemistry, but that's pretty freaking cool. I know like the basics, so I can understand stuff, but I don't actually know chemistry. Synthesized from choline. Acetylcholine, for example, is synthesized from choline, which is abundant in milk, eggs, and peanuts. So if I have a ton of that, the amino acids phenylalanine, phenylalanine, the amino acids phenylalanine and tyrosine present in virtually any protein are precursors of dopamine, norepinephrine. Precursor, phenylalanine. The amino acid tryptophan. Precursor to serotonin. Serotonin being one of monoamines. Serotonin being the endoleamine. Tryptophan, precursor to serotonin, crosses the blood-brain barrier by a special transport system that shares with other large amino acids. The amount of tryptophan in the diet controls the amount of serotonin in the brain. So tryptophan is good then. So your serotonin levels rise after you eat foods richer in tryptophan, such as soy, and fall after something low in tryptophan, such as maize, American corn. However, has to compete with other more abundantly large amino acids, such as phenylalanine, which is a precursor. My brain is literally breaking. Tryptophan, serotonin, phenylalanine, and tyrosine, precursors for dopamine, norepinephrine. Okay. Phenylalanine, tryptophan, serotonin. Foods rich in serotonin and tryptophan raise serotonin levels, such as soy, and fall after something low in tryptophan, such as maize. However, so sometimes you may not get the tryptophan if you have an intake of larger amino acids, such as phenylalanine. One way to increase tryptophan entry to the brain is to decrease consumption of phenylalanine. Another is to eat carbs. Carbs increase the release. Of insulin, a hormone which takes several of the competing amino acids out of the bloodstream and into body cells, thus decreasing the competition against tryptophan. So is that to say that if you have more phenylalanine, I feel like I'm confusing dopamine and serotonin. Dopamine. So dopamine is the feel good. Okay, what is serotonin the sleepy one? I think it is. Serotonin is the sleepy one. Mood stabilizer. Okay, no, wait. Think of it in terms of sleep. What do you take to make you sleepy? I can't remember. Can you guys remember? What makes you sleepy? Okay, wait. I literally did a video on this, and I can't remember. Melatonin! Okay. Oh my god, that took me forever. So dopamine, serotonin, melatonin. Melatonin is the sleepy one. Dopamine excites you, and serotonin relaxes you. Serotonin is used as a mood stabilizer. High levels of serotonin make you feel agitation, restlessness, or irritability. Affects mood. Okay. I thought dopamine makes you dopey. At least for... But dopamine is also considered a feel-good hormone. Feel-good what? Feel-good neurotransmitter. So serotonin is like the bad excitement, and dopamine is like the good excitement. That's kind of what I'm reading. Because high levels of serotonin lead to agitation, increased heart rate, restlessness, which doesn't sound that good. And then dopamine is just like you feel good. Interesting. Okay, let me read this again. Norepinephrine, epinephrine, I mean, like all of those things are used. Wait. Isn't epinephrine the stuff that they use to like revive you if you die? If you know what these things are, good for you. Because apparently I don't. Adrenaline! Yeah, epinephrine, adrenaline. Push whatever cc's of adrenaline. No, epinephrine. Okay. Used to treat life-threatening low blood pressure. Used during CPR. Okay, norepinephrine is one that they use for cardiac arrest, and then epinephrine is just another word for adrenaline, but they're both a form of adrenaline. Okay, let's read this again. I know this is taking me forever, but it's worth it. So amino acids, phenylalanine, tyrosine, precursors for dopamine, the feel-good hormone, norepinephrine, the amino acid tryptophan, precursor to serotonin. Okay, the amount of tryptophan controls the amount of serotonin in the brain. Serotonin levels rise after foods rich in tryptophan such as soy, meaning that you are more alert, but could also lead to feelings of restlessness and high. Yeah. Oh, interesting. Okay. Fall after something low in tryptophan, such as maize. So, things with high carbonates. Let's see. However, tryptophan is can be large amino acids such as phenylalanine, which is carries dopamine and adrenaline. One way to increase tryptophan entry to the brain is to decrease consumption of phenylalanine. How do you decrease phenylalanine? Another is to eat carbs to increase tryptophan. So when you eat carbs, it increases the release of insulin, right? It takes several of those amino acids out of the bloodstream and into body cells, thus decreasing the competition against tryptophan. So what happens to those the dopamine, epinephrine? Because carbs are used as energy, adrenaline is a form of energy. Increase the release of insulin. To eat carbs. Carbs increase the release of insulin. Okay, now I want to relate this back to like diabetes, because how does that work? Because insulin, when you're not getting enough insulin, or you're getting too much insulin, I don't know what the difference is. But yeah, if you eat more carbs, then the bigger amino groups will be, and there's more room for tryptophan, which means you'll have more energy. Sure, that makes sense to me. So, phenylalanine from the diet, tyrosine, dopamine, norepinephrine to epinephrine. Tryptophan from the diet, hydroxyapatite, deoxytryptophan, the serotonin, acetylcoenzyme. We have choline, and then you get acetylcholine. Huh. Okay. I need to snip that. That's important information. Okay, now we're getting into transport and storage of transmitters. It's abundant percent size in the presynaptic terminal. However, the neuropeptides are synthesized in the cell body. Neuropeptides. So amino acids in here. And like monoamine and acetylcholine here. Neuropeptides in the cell body transported down the axon or into the dendrites. Unlike other transmitters, neuropeptides are released from many sites in the cell. Okay. Speed of transport varies from only 1 millimeter per day in thin axons to more than 100 per day in thicker ones. And this is talking microns. Okay, presynaptic terminal stores high concentrations of neurotransmitter molecules in vesicles, tiny clear spherical packets. Yes. Nitric oxide, the gaseous neurotransmitter mentioned earlier, is an exception to this rule. Neurons release nitric oxide as soon as they form it instead of storing it. So that's like a continuous process. And the neuron is like the whole thing. It's one axon to one neuron. So that's like the presynaptic terminal also maintains much neurotransmitter outside the vesicles, so there's stuff inside and outside. Anatomy of the synapse. Cerebellum as well. Vesicles. I can't even tell what's what. Okay. In some cases, neurons apparently accumulate excess levels of neurotransmitters. Neurons that release serotonin, tryptophan, that release dopamine, alanine, or in norepinephrine containing enzyme MAO, monoamine oxidase. Enzyme is like a energy trigger thingy. Wait, do I know what an enzyme is? I'm like trying to review all of the biology I know, and it's been a couple of years, y'all. A couple of years. Enzyme. I know that has something to do with some kind of energy. Proteins. Catalyst. That's what I meant. Energy. The catalyst. Enzymes are catalysts. Yes. So catalysts are like that spark that you need to get a chemical reaction going. Yeah. Okay, so let me read this again. Neurons to release contain an enzyme, which breaks down these transmitters into inactive chemicals. Huh. Yeah, because they turn into inactive chemicals in order to be within the synaptic cleft. Then once they are absorbed by the postsynaptic neuron, postsynaptic whatever. What is it called? Yeah, postsynaptic neuron. Okay, that's right. Then that's where they get their polarization again. They get their charges back. So like They go through this process. That's the ionotropic metatropic thingy. The EPS. No, not that. It's this. The ionotropic and metabotropic receptors are like these changing rooms that change ions with charges into chemicals that have no charge or inactive chemicals to be transported in the neurotransmitters. And then once they're back at the postsynaptic, then they go through a change again in which they get their ions back in order to be transported down the other axon / neuron. We shall return to monoamine oxidase, and discussion of depression because several antidepressant drugs inhibit these enzymes. And the enzymes trigger the release of serotonin, dopamine, norepinephrine. Inhibit norepinephrine. Oh, because if you have too much of dopamine, norepinephrine, of adrenaline, it tires out your system. And I think that's related to decreased mood. I think that's why. So if they inhibit this enzyme to release more of the serotonin, to get that to create those in excess, I think it's just to balance it, because you need a balance. Basically, you're out of balance. Like if you have a disorder, you're out of balance. And I guess medicine is trying to keep you in balance. I don't know if that makes any sense. I know I haven't even taken a break. I don't know how long I've been streaming. Oh, an hour and a half. I should probably take a break because it's been an hour and a half. So I'm gonna go ahead and do that. I should be back in like 5 minutes, just giving myself some time to decompress. And if you're new here, if you're watching, I'm just studying, and you're welcome to study with me. It's literally all it is. You're welcome to follow, subscribe, like, comment, anything. But yeah, I will be right back. So I'm actually gonna eat some dinner. I don't know if you guys want to hear me chew. If it bothers you, let me know. If anything, it's like a mukbang and we're just eating all together. If it does bother, I will just switch off my mic. But if not, then I'm gonna keep eating. I'm eating quesadillas and black beans. Because why not? So that's interesting. This entire time I thought that after the action potential in the axon of the neuron was released, honestly, I thought there were like a ton of neurotransmitters that were released, but apparently it's only a few, like 2, 3 at a time. But then this one's saying that there could be a combination of different neurotransmitters that are released at 1, 2, 3 times. So that's kind of confusing. Oh, I should eat like this, but it's hot. I haven't even worked on problem set 2 yet. I'm still on brain and behavior. Apparently that messes with my live stream, so I'm gonna do that. So that was from the YouTube. Interesting. Yeah, I think I'm gonna focus tomorrow more on the rest of this. And then I'm gonna move on to, I need to do my video for my video class, but that involves using programs on my computer that would overwhelm the CPU and therefore mess with the live stream. So I'm gonna try to work on my research with stats class that also involves using a program. The assignment for that is due soon, so I want to work on that as soon as I can. And I believe I had problem set 2 already open. Did I finish this? It looks finished. Yeah, this is... What does it mean problem set 2? Oh, did I get the answers? already turned in problem set 2. I was waiting for problem set 3. Hold up. I need to check this. I'm pretty sure I already turned it in. This guy had me freaking out about problem sets. But I think someone already posted that they haven't released them. What? Oh yay, I got full points. Problem set 2. I already submitted, yeah. Okay. And then problem set 3 hasn't been posted yet. So this is discussion answers. And this one is correlations. This was the DAC. This is what we went over during class. But it's not an assignment. Okay. Okay, so I'm all caught up for this class now. wait. I hope they'll give me an extension. I have tomorrow to meet. Oh, and I'm free Friday again. Friday is off. That's cool. Let me go back to this one. Yeah, 10 to 12. But I use that time for other stuff. And it's Saturday before 2 p.m. When is that document due? I know it's due soon. Oh, okay. For some reason I thought it was due on the 19th. Okay, so it's not due soon. For some reason I thought it was due the 19th. Okay, so I have time. Oh, that's happening. That's on the 19th. It's tomorrow. Okay. Here, probably gonna do that tomorrow. Thanks for the heart. I was like totally absorbed in that email. It takes so long to send an email because you want to word it right. But basically, I'm going to be learning about research that's being conducted in regards to microaggressions and their impact on cognitive abilities. So that's cognitive functions like, I don't know, it could be, what could it be? Well, everything cognitive, the way your brain works. But I think it's in terms of how well a person works cognitively, meaning, can't think of the word, critical thinking, and stuff like that. I can't even think right now. But yeah, so that's happening. I know I have stuff to do for honors. I usually have stuff to do for honors. Be fixed by Tuesday. I think this is so dumb. Be prepared to discuss in lecture and discussions this week. That's why I said last week about the video. We didn't discuss. We didn't discuss anything from the usual video because the professor who's leading the class, like none of them are on the same page. Basically, they don't know what the other is doing, and honestly, I think it's very unprofessional. But you know, especially because it's the honors program. Like, I would expect a little more structure, basically. Celebrate exercises. Oh, I probably have an assignment. I think it was due yesterday. No, she extended the deadline. Oh, praise the Lord. Okay. I was about to panic. Just do tomorrow. I'm gonna do it today. I'm basically done with it. I literally almost had. That's why I didn't like freak out about it yesterday or this entire weekend because it's not due till tomorrow. Although it's due at 8 a.m. of all times. Okay. Still says something. Awesome. Wait. This was a long document. She didn't even. They didn't even post week 2. She did say she was stressed out though, so that makes sense. Okay. So, honors is good. I need to work on the video. And that's due tomorrow, so I'm gonna work on that tomorrow. Psych, I already turned in the assignments I need to turn in. And then I have an exam, not this week, but next week on the 29th. And so far, I feel prepared. I really don't feel that bad. So I think I'm gonna just keep studying brain and behavior because that takes so much time to study. But I know it's gonna be worth it. It's gonna pass those exams. Oh, it's a question. I think that I should do a video on how I study and how much time goes into how much I study, and also what my study methods are, and then what programs I use to study. If that sounds interesting, let me know, and I'll try to record a video and I'll post it on my YouTube. And I don't know how to post that to Twitch, but I'm pretty sure I could post it to Twitch. But if that's interesting in any kind of way, please let me know, and I will make one. Write it in the comments, write it in the chat, and let me know. Thank you. I mean, good on her. So I get updates from the New York Times on my phone, And there's a lot of live updates about the US presidential transition because guess what? Biden's going to be sworn in 2 days. Yes. I'm really excited. And so they've been reporting a lot of updates on like people basically people getting caught for raiding the Capitol when that whole thing happened. And so this woman from Pennsylvania, accused of taking Nancy Pelosi's laptop, turned herself into the police. And I'm like, well, good on her. I mean, you could argue she took too long, but honestly, that kind of thing takes time to decide to do. And I'm just grateful that she decided to do it all together. I don't know anything about the woman, but people are crazy. I think I should spend some time reviewing because I still need to add more questions. So this is active recall. You're gonna hear me talk a lot. I'm so sorry. My goal, I don't know if you could see this scale. It's from red, orange, yellow, blue, and green. My goal is to get all of these to be green. Right now, a lot of them are blue. It really takes a lot for me to say that I have something green, because when it's green, that means that I could just say it without even much thought. Like it comes up. Blue it takes me a while to get to the point, but I know, and so on and so forth. Red, I just don't know it at all. I'm just like, what is that? This is going back to the very beginning. Neurons and glia. Cool. Levels of analysis. What is the process behind our behavior? I have my own definition that I've made into the definition. So basically, the process behind our behavior. So our behavior comes from stimuli from our external environment. So that's everything from the light, my humidifier going off, shapes and colors and everything, external stimuli, and then internal stimuli. So that's everything that's happening within my body. So what is the process behind our behavior? So behavior is a response of that stimuli, and that stimuli needs to be processed and integrated in our minds before becoming an action or a response. So that is our behavior. Did I say it exactly as I wrote it? No. Integration, good. And actions based on this processing. So keywords: processing, integration, intake. Taking in, intake of internal and external information, stimulus. Perfect. I can give myself a green with that one. I feel good. What is the neocortex? Neocortex. It's basically, imagine the brain, the organ, the brain, and it's the general area of the brain that's like the outer part of an orange. So think of an orange, you have the juicy flesh inside, and then you have the rind on the outside. That is the neocortex of the orange, the rind, the skin. That is exactly what the neocortex is of the brain. It's a region of the brain, the external area, and that is the neocortex. What's it called? I mentioned that actually during the Zoom in class, and then my professor was just like, exactly. And I'm like, yes. I'm glad our brains think similarly. But the external region of the brain. What are neurons? This is never what I think it is. What are neurons? Neurons. Okay, neurons are. Wow, I don't know what it is. I I really don't know. And I have a blue on this. That's horrible. I'm making you an orange. I'm pretty sure that's what it is. Neurons, they're messengers within the brain. Create synapses that signal transmissions along axons for long distances. Watch this not be it. Wait. How do we get the neurons in the brain? What are glia? I can tell you what glia are. It's never what I think it is. Okay, glia are, we have the astrocytes, the oligodendrocytes, Schwann cells, microglia, and that's it. It's those four. Astrocytes, oligodendrocytes, Schwann cells, microglia. Those are glia. Glia are they bind to that's astrocytes. I'm talking about astrocytes. Glia are, they're kind of like the janitors of the brain. They help to modulate synapses, synaptic transmission. They help with that synaptic transmission. Glia, oh my god, glia. They're a support metabolic role within a neuron, or not within a neuron, but that works with the neuron. Support metabolic role. Watch those be the general properties of glia. Okay, fine, I'm opening it. I don't know what it is. I'm so upset. I literally said that, but okay. What are neurons? Receive information. Transmit it to other cells. I said transmission, I didn't say receive. I'm still leaving it at an orange because that took me forever. What are glia? They do not transmit information over long distances. They do exchange chemicals with adjacent neurons. Yes, that has to do with the processes in the the synaptic cleft with adjacent neurons. Yes. I did mention the different types. I'm gonna leave you at yellow because that was horrible. Wow. The last time I opened this, I should add a property. It tells me created time, last edited time. Yeah. Last viewed. Oh, that's literally it. Oh, because I changed the colors. I wonder. No. Okay, just leave it alone. last time? This was last Tuesday. Last time I studied this was several days ago. That makes so much sense. That's really bad. I don't have a calendar for the days and times that I should be reviewing stuff. Okay, cells of the nervous system. Moving on. Cells of the nervous system, neurons and glia. How do we count the number of neurons in the brain? Okay, so there's no easy way to count all of the neurons in the brain because even from person to person, we have a different amount. And then within the regions itself, within the brain, there's different amounts of neurons in each part of the brain, and they're not like all evenly spaced out. So it's difficult to count them. Like you can't even just take one small region of the brain and then count those and assume that that's how many there is in the entire region of that brain, because they're not evenly spaced out. And some regions of the brain have more neurons than other regions of the brain, which is crazy to think about. So how do we count them? So the way scientists have done it, you have to take multiple, like a lot of samples basically, and once you've taken a sample from one region of the brain, you have to do a process of suspending them in a fluid and then homogenizing them, meaning spacing them out evenly, and then, splitting that in half and counting this one part and that one part. It also involves staining the neurons so that you can actually tell which ones are neurons apart from everything else in that fluid that you took out of the brain. It's a whole thing. It takes a lot. So that's how do we count neurons in the brain. So then it comes to the question of how many neurons are in the brain. And there's like 3 different answers. Okay, it's just baffling to me. So you have the textbook answer, the widely accepted answer, and then you have the most probable answer. Most probable answer is that there's about 85 billion neurons and 85 billion glia. But although they do say that there's far more glia or glial cells than there are neurons. So I don't know about that either, but that's what they say, 85 billion. Now for the textbook answer, there's an assumption that there's about 1 trillion neurons to about 10 trillion glia. And then the most widely accepted answer says that there's about 100 billion neurons and about 10 trillion glia. Those are my numbers. That's what I'm sticking with. I could be wrong. Let's see what I wrote. Okay, so I had them backwards. The widely accepted answer is the 1 trillion neurons to the 10 trillion glia. The textbook answer is 100 billion neurons to about 1 or 10 trillion glia. Yeah. But most likely it's 85 billion neurons glia respectively. Okay, I'm giving myself a yellow because I actually did pretty freaking good on that. Those are crazy numbers to memorize. Okay, moving on. Types of glial cells. This is what I was trying to get at. What are the general properties of glia? Keep it. Oh, I'm putting this little acronym KISS. Keep it simple, stupid, on these two because I'm trying to answer these in like paragraphs. But no. Okay. What are the general properties of glia? There are three. They are metabolic support functions. They help with modulating synaptic transmission, and then they're kind of like our janitors. Yeah, something like that. Let's see what it says. Fuck. Sorry, I'm so sorry. I got 2. Okay, there were 4. So uptake, release, modulate synaptic transmission. Yes, got it. Work as supportive roles, metabolic roles. Got it. They are non-conducting. They have no charges. They are not ions. They are not acids. I mean, they're not neurotransmitters. Those have charges. And then they divide as adults. That's really important, because as we learn later, neurons, if you lose them, you lose them. They don't, you don't regenerate neurons, which is crazy to think of. But glial cells, on the other hand, if you lose some, they'll make more, which is interesting. So they divide as adults. Those are very important properties. Okay, I'm gonna put a 4 here because for some reason I can't remember. And I'm gonna make this a yellow because I got half. Okay. How do astrocytes interact with neurons? That's cool. So one of them is modulate synaptic transmission. Astrocytes, they bind to capillary walls, blood vessels, to synapses. They also help with reuptake of neurotransmitters, astrocytes. They also absorb, I want to call it waste, neural waste, dead neurons, dead everything. Very similar to microglia. Microglia is similar. They help with the immune response, and they help with primarily inflammation within the brain. They're also like the janitors of the brain as well. They pick up a lot of the dead material. Astrocytes do as well. Yeah. Okay. Crap. Yes, they work as scaffolding for neurons during development. I know this. Because they bind to stuff, they attach to them. They allow neurons to attach to them too, so they work as like this, like they help keep stuff in place when the brain is developing. They control the amount of blood flow to each area of the brain. Yeah. They attach capillaries, neurons, same thing, synapses, blood vessels. Remove waste material when neurons die. I got that. Modulate synaptic transmission, uptake release of chemicals, including neurotransmitters, synchronizing the activity of axons. I got like half of that. I'm gonna give myself a watch me come back to be like, crap, that's right. Make these orange again. Nope, I'm keeping them orange. I don't know them well enough to be yellow. Okay. What properties differentiate oligodendrocytes and Schwann cells? What is their role in a neuron? This one I know. So oligodendrocytes and Schwann cells both produce myelin. Myelin are those little round tubule thingies that wrap around axons, and they help basically with speeding up the action potential. But the difference is oligodendrocytes are the myelin found within the central nervous system, the CNS. The Schwann cells are the myelin found in the peripheral nervous system, the PNS. What is their role in the neuron? I already said it. And yeah, that is that. Oh, well, I didn't say that, but okay. Regeneration. Myelin, yes, myelin has to do with regeneration, which has to do with the bad glia, which I'll explain later. What is their role in the neuron? Yes, uptake release of chemicals, including neurotransmitters. All of them do that. And then modulate synaptic transmission. All of them do that. Okay. I'm gonna keep that blue because I didn't say the other stuff. What are the properties of the microglia? I already said they help with inflammation in the brain, so having to do with, what was that called? Let me think. Okay, I know it has to do with white matter. Bundles of microglia is white matter. Bundles of axons, God, I have to study that later too. Bundles of, not axons, but that constitutes gray matter. They pick up the white matter. So white matter spots in the brain. So if you see an image of the brain, there's a bunch of it's usually gray. And when you see spots of white matter, that's dead brain cells. Okay, so microglia. This is what I get for not studying this for like a week. Oh my god, what are the properties of microglia? Microglia. I'm like studying all this other stuff. There's no room in my brain. I know the general population. Let's just talk about microglia. Okay. They help with inflammation in the brain. It's white matter. White matter is involved somehow. I just don't remember how. And then they pick up neuronal waste. Yep. It modulates synaptic transmission, obviously. That's all I can remember. Immune response. That was the word I was looking for. Help to clean up debris. Yeah, neuronal waste. Oh, I should remember that though. They also help with viruses, fungus, and other bad stuff. They're like vacuums. They eat it all. Nowhere on here does it talk about white matter. White matter must be somewhere else, but I know it's there. So immune response in the brain. I know why I keep thinking white blood cells. White blood cells is the body's immune response. Microglia is the immune response in the brain. Okay. Response to inflammation. Uptake, modulates synaptic transmission. CNS. Yes. I never looked that up, so I'm just gonna leave it alone. Okay. I'm gonna leave that blue because I basically said all those things. I just didn't know. How did bad glia behave? This one I know. So, okay. So, where do I even begin? So remember when I was talking about regeneration of cells. So, glia in the brain inhibits the process of regeneration in the brain for some reason. If the neurons, how do you say this? For example, in the external areas of our body, the peripheral nervous system, so nerves in our arms, nerves in our legs, things like that. Or maybe not our legs, because that can be connected to the spine. Anything that is not connected to the spinal cord in the brain is basically the peripheral nervous system. So if you were to damage any of the nerves or connections within those areas of the body, you can potentially heal and get better. You can regenerate those lost cells. Now, anything within the spinal cord and the brain, bad glia or glia inhibit that process and don't let it regenerate. So when you have injuries to your spinal cord or injuries to the brain, that microglia inhibits that from recovering. So if you have an injury to your spinal cord, you're paralyzed and nothing can basically change that. But what they're hoping to do is to use the, not the glia, it's something else in the peripheral nervous system to take those cells, kind of like stem cells, and use them as bridges within the spinal cord in the brain to help that process of regeneration to heal people, basically. So that's how bad glia behave. So molecules in the CNS don't regenerate. Glia inhibit that regeneration. They want to improve the CNS, spinal cord in the brain, regeneration by using the PNS tissue as a bridge in order to promote the growth. So to block the bad glia and to promote growth. Yes. I'm going to give myself green. Yay. Right, now we have the plasma membrane. What kind of activity takes place in the plasma membrane? The membrane is bilayer, meaning that there are hydrophobic phospholipids and the hydrophilic tails of the phospholipid. Hydrophilic meaning that they love the water, they attract water, and then hydrophobic meaning they expel stuff. So the main purpose of the membrane is basically to keep things out and also to let certain things in. That's the main activity that takes place in the plasma membrane. The membranes within the brain are different from the membranes in other areas of the body. The membranes in the brain are like really tightly knit, and like it's very difficult for things to go in. It's also very difficult for things to get out because the membrane is just really tightly packed. Other membranes are a little bit more loose and they potentially let stuff in and out.
9 o'clock.
And yeah, so that's a plasma membrane. But bilayer, yep, allow things in, allow things out, channels, receptors. I don't really talk about that, but if I see it, I would remember it. All right, I'm gonna make that green. Okay, the axon. What is contained within the cell body? Cell body, yeah, the cytoplasm, the nucleus, the axon's DNA. That is the cell body. Yeah, cytoplasm. Eos, nucleus, DNA. Wow, for the first time ever, I got all of it correct. Good job, Nance. Oh, sorry. What is another name for cell body in its plural form? Another name is called somata. Somata. And then its plural form is... Oh, wait. The name for the cell body is soma. Plural form is somata. Yeah. Alright, cool. What are the properties of an axon? Okay, the axon consists of the cell body surrounded by dendrites, axon hillock, the axon itself, and then the synaptic terminals. And on the axon, there could be myelin. And then the nodes around here are the space between myelin. What are the properties of an axon? Oh, they allow transmissions over long distances. Yeah, transmissions of information throughout the brain from one axon to another. There's only one axon to one neuron. Watch me get this right. Ooh, I'm excited. Yes! Yeah, I didn't talk about the axon hillock, but that's fine. Long distance transfer output of info. Yes! Oh my god, I got it right. I'm so proud of me. I'm gonna make you blue. Yeah. Let's see, hopefully I remember you next time. What are collaterals? Oh, crap, that's new. Collaterals. Collaterals. What are collaterals? Let me look at this. What are collaterals? Collaterals have to do with the axon. Collaterals have to do with the axon. But what did they do? What did they do? My brain. I want to say that's what collaterals are, but I don't think so. Transmissions that don't trigger the action potential. I'm pretty sure that's not right. I should drink more water. Branches of the axons. What? No. Ooh. Okay, first of all, why are you gonna tell us that and then not invite us to go? I mean, I guess people like to. What are the properties of an axon? What are collaterals, branches of axons? Branches of axons. Axons. Wouldn't that just be the dendrites? Okay, I know this is in the textbook. Let me. I just realized you guys actually don't hear me chew. That's crazy. Am I spelling it wrong? Oh, this has to do with plasticity, so that's why we didn't talk about that much. Collateral sprouts is the actual name. Branches of axons. So I guess collaterals in a way, or collateral sprouts are in their own way a process by which the brain tries to compensate for lost neurons. I'm getting really distracted. Okay. I'm going to keep that at red because I had no idea what it was. What occurs at the axon terminals? A lot of stuff occurs at the axon terminals. Axon terminals. Information is definitely transferred. I don't know. Okay, contains vesicles of the neurotransmitters. Communication between cells. This communication leads to a synapse. Yeah, I mean, I knew that, but I guess I was just being lazy. I'll leave it at yellow. What starts the process of myelination? What does it help with, or how are the nodes of Ranvier involved? Alright, so what starts the process of myelination? Oligodendrocytes in the CNS and Schwann cells in the peripheral nervous system. What what does it help with? So the myelination of the axon basically allows us to move throughout the axon. There's a constant need for the action potential to push the transmission through the axon. What the myelination helps with is basically it takes less work for the synapse to send the signal down the axon. And the nodes of Ranvier are involved is because the nodes of Ranvier are where the synapse jumps across the axon to reach its landing quicker. That's basically what it is. Increase action potential, conduction, and velocity. That's basically what I said, just not in the same words. And then they are the breaks in the myelin sheath. So they make it easier because I don't really know how to explain that. What is a synapse? I'll make this yellow. What is a synapse? A synapse is it's an exchange of information. No, synapse. Action potential. It's the translation of the action potential into a message to be sent to the next neuron. This is not the synapse, that's a synaptic cleft. A gap between the axon terminal and the dendrite. Of another axon. It's a place of chemical communication where neurotransmitters are. I'm gonna make this blue because I actually knew what that was and that wasn't the right term for it. I'm gonna make this blue as well because I knew the majority of it. What occurs at the axon terminals? Neurotransmitters are released. What happens at the axon terminals? Neurotransmitters, vesicles are sent there with neurotransmitters, and it's an area where neurotransmitters are released. It's a space. Yeah. Let's see. That's where communication occurs. Axon terminals, yeah. What are collateral sprouts? Collateral sprouts are basically growths of an axon extending itself to other neurons. What are the properties of an axon? So the properties of an axon is that there is one axon to one neuron. Axons transmit and receive information over long distances. Axons, the axon hillock is where the action potential starts in order to become an action potential, and then just one more properties of an axon. Oops, we're so dry. What are the properties of an axon? I named a handful of them. Oh, I said every single one of them. Right. What is contained within the cell body? The cytoplasm, the nucleus, and the DNA within the nucleus. Within the cytoplasm is processes of protein synthesis and other protein actions. Yeah. What is the purpose of the synapse or the synaptic cleft? It's a space between the axon terminals and the dendrites of the next neuron. It's a place of communication or transfer of neurotransmitters. Purpose of the synapse. So we have the synapses. The synapse is just an area of communication. In the same way, the axon terminals are an area of communication. starts the process of myelination. Myelination is started by Schwann cells and oligodendrocytes in their areas respectively. Myelin helps the action potential travel faster down the axon, and then the nodes of grandia are the gaps between the myelin. Okay, let's go back up here. What are neurons? Neurons are a vehicle of communication between neurons. Watch me get this wrong. Receive information, communication, receive information, transmit information. That's true. They receive information, then transmit that information. Yeah. Glia. There are two things to that. The glia. The glia are all these things. They help to modulate synaptic transmission. That's intake and uptake of the transmitters. And help with transmission. That's right. They don't help with long distance. They don't the glial cells don't travel long distance. What's it called? And they have no charge. They're uncharged. Yeah, they are uncharged. That wasn't in there, but okay. So that was this one. Familiarity. I'm gonna say okay. Studied yet, but I've written down questions. Okay. Terminology. Groups of neurons. Oh, this is the one I was talking about. What are the plural terms in the CNS for cell bodies and axons? What are the plural terms in the PNS for cell bodies and axons? Crap. Cell bodies. I think I remember a term, but I guess it's for the penis. Cell bodies. Groups. Plural term for cell bodies. I cannot for the life of me remember. Okay, I remember something about a bundle of tracks. Tracks, I think it's for the penis. Bundle of tracks leads to gray matter, and then bundle of axons. I don't remember. Cell bodies are nuclei. Nucleus, nuclei, cell body, cytoplasm, soma, nuclei, CNS, brain, spinal cord, nuclei, nucleus, multiple nucleus is nuclei, cell bodies are nuclei, bundle of axons, I was right, oh, I kind of said it, just didn't know that's what it was called, white matter, I was right about that, bundle of axons are called tracks, bundle of tracks is white matter. Okay, not too bad. I'm gonna put these as orange. Okay, what are the plural terms in the PNS? The image of the word is like at the very edge of my mind. Cell bodies, PNS. seriously glands, something G gland. I don't know. Ganglia. Like I said, tip of my tongue. PNS nerves. Bundle of axons is called nerves or white matter. Bundle of nerves in imaging. Yeah. Okay, so then those. Okay, so I said I need to correct myself in something that I said earlier about white matter, brain scan, white spots. That's not. I mean, technically in a scan, that's dead space. But this is different. This is what I was thinking. White matter, bundle of nerves in imaging. Maybe it is. I could be wrong, because those are bundles of axons. Maybe they're dead axons. I don't know. But I totally did not know this one. That is for sure red. Where can nerves be found in the nervous system? Where can nerves be found in the nervous system? Nerves, the PNS? Isn't that what we just talked about, the PNS? Oh my god, yeah. Okay, this will be easy to remember later on because we're going to talk about these. They can be sensory. Nerves can be sensory, motor, or sensory motor. To carry things in and out of the nervous system. Nerves. Yeah, I didn't know this one. Okay. Blood-brain barrier. I can talk about the blood-brain barrier like the back of my hand. Let's see how well I do. What is the main job of the blood-brain barrier? Okay. Main job of the blood-brain barrier is to keep things out of the brain and to keep things inside the brain. Literally, it's just to keep stuff out. It's to protect the brain. Protect it from toxins, from viruses, from everything, good or bad. Yeah, that's literally the job. Controls the access of molecules to and from the brain. Yeah. I'm gonna put that as yellow because I talked too much. What is the difference between the blood-brain barrier and other membranes? I talked about this earlier too. The blood-brain barrier, the membrane is very tightly packed, like very tight junctions. Yeah, compared to other membranes, which can be a little looser, let things in and out. The blood-brain barrier does allow some things to just go straight through. I mean, that's everything from, I think, like oxygen. I think it's inorganic compounds. Stuff that's organic, like stuff with carbon, is a no-go. I think that's what I remember. Interfere cells of the wall of the blood-brain barrier are more tightly joined than other walls where there might be small gaps. That's basically what I said. I'm gonna make that yellow. Why is the blood-brain barrier so important? It's important because of exactly what I already said, blood-brain barrier. It's very difficult for things to get in, for things to get out. It's literally it. Yeah. I said this keeps out the bad stuff and keeps out the good stuff too. But this one I didn't say, so I'm gonna give myself an orange because that's actually a really important point. So the blood-brain barrier is very important because, as we have mentioned before, neurons, you only have so many of them. They don't continue to divide or make more of themselves when they're adults, when they're fully grown. As as compared to other cells in other area of the body that can regenerate, like our red blood cells, what's it called, mitochondria, and like all those cells, like all of that stuff can, like, you can make more of that. They can continue to divide as we grow older, but neurons, no. They don't continue to divide. So it's very important to keep stuff out. It's very important for that blood-brain barrier to exist, as like a very, very strict TSA. But yeah, that's basically what it is, TSA. Except it's not, what's that called? Compromised. It's not, there's a word for that. Corrupt. There you go. It could be though. That's why you have neurodegenerative diseases and whatnot, right? Be corrupted. Neurons don't regenerate. Yeah. So we don't want to let cells in the brain get compromised. Yeah. What can get through the blood-brain barrier? So that's what I was talking about. There are some things that could just like go straight in. But I'm not sure. I don't remember what they are. Small uncharged molecules, including oxygen and carbon dioxide. Okay, so not not organic molecules, uncharged molecules like oxygen and carbon dioxide. Small uncharged molecules. Okay. Water crosses through small special pressing channels in the walls. Water makes sense. Molecules that dissolve in fats. That's right. Can cross passively, so that's important. I guess I'll talk about it later. Why are these difficulties so important? The difficulties for stuff to pass through the brain barrier. We've already talked about that, but it's easy. It's easier for stuff to cross the membrane if they are binded to a fat. Yeah. So, for instance, a lot of medications that treat mental disorders, mental illnesses, it can be very difficult to get them. What's that word? To make different ones because it might work in the lab, like the thing might do the process it's supposed to do, but then when it comes to the blood-brain barrier, if it's not attached to a fat, then it's difficult for it to even get inside of the brain. That's basically it. So we need it for medications. That's important to know. Okay, so this one I did not know at all. I mean, I knew, but I didn't know. So I'm going to put it as red. How does the active transport help other essential chemicals cross? So active transport is basically a carrier, a messenger. It allows things to get in. Oh, it's a lot of information. I don't know if you can tell, but my brain is kind of getting really sluggish, and I really don't want to. I think I'm going to start with this next time, because yeah, my brain is kind of like doesn't want to work. Although I have been studying for how long? 3 hours. Yeah. Thank you. How does the active transport help other essential chemicals cross? First mediated process that expends energy to pump chemicals from the blood into the brain. Okay. Essential brain chemicals cross. Essential brain chemicals cross. So glucose, right? So these are things that can get pumped into the brain. Glucose, amino acids, purines, choline, a few vitamins, certain hormones, iron. I don't think I tried memorizing these. I think I just did straight. 1, 2, 3, 4, 7. There's 7 of them. 7. Okay. Glucose makes sense. Glucose. Mentioned purines. I know what those are now. Choline. Also know what that is now. Amino acids. Also know what those are. Certain vitamins, hormones, and Iron. Iron is a mineral. Okay. Amino acids, purines, choline. I already forgot. Glucose, which is a sugar, which is it I don't think it's a hormone. Sugar, glucose, iron, some vitamins, and certain hormones, right? Okay. Protein mediated process. Protein mediated process. Protein. Why protein? That expends energy to pump chemicals from the blood into the brain. Protein. Why protein? Why protein mediated? So that is an active transporter. That's definitely a red. I have no idea what that was. Red. Why are these difficulties so important? I feel like I've already said this. This has to do with drugs. Okay, then I will give myself the orange. Okay, I'm gonna move those because membrane potential of cells. What are the properties of membrane potential? Membrane potential. Membrane potential. What the heck is that? Membrane potential. Membrane potential. I think this is going back to, what is the difference between a membrane at rest versus in action? What is the refractory period? Yes, it is talking about that. Membrane potential. My brain is so fried. Okay, my brain is fried. It's been 3 hours. I'm gonna take a short break. I think I'm gonna give myself like it's 9:30 right now. I think I will be back at like 9:40. So give me like 10 minutes because my brain is fried. Like I can't. Okay, so I will be right back. anything sticks. Membrane potential cells. So properties of membrane potential. Properties. Membrane potential. Okay. Oh, yes. Okay, I remember. So the membrane is depolarized. So that means that, no, the membrane is polarized. The process is depolarization or hyperpolarization. It is polarized. The membranes of the neuron tend to be at about -65 millivolts. And so in order for it to be polarized, basically the charge needs to reach closer to 0 in order to activate an action potential. And wow, that break really helped. And in order to activate the action potential, it needs to depolarize, which means to get closer to 0 in order to reach a threshold where the action potential actually becomes something, or the potential of the membrane reaches an action period called action potential. So what are the properties of the membrane potential? There you go. So what is the difference between a membrane at rest versus in action? A membrane at rest, right? Oh, that's the other thing too. A membrane at rest, it has a voltage gated channels, and so you have it at rest. There is a certain amount of potassium inside, more so than there is outside, and there's a larger amount of sodium outside than inside. And so when an action potential is triggered, sodium rushes in, right? Because the voltage, the depolarization of the cell opens those voltage gated channels, lets the sodium rush in, and that triggers an action potential. Oh, my brain is working. Okay, what are the properties of membrane potential? Hopefully I said everything I needed to say. And it's empty. Oh my god. Let me guess. Empty. Let me guess. Empty. So I never wrote anything. Okay. I'm right either way. I'm right. Oh yeah. Let me exit out of that. Let me open this. I need to talk about neurons. No, that was something else. Here. Okay. That's been bothering me. Here it is. Yes. Oh my god, I remembered all of that. I feel so great. refractory period, and you have what's that other term? It's absolute and something. So refractory period and then absolute refractory. There's two. That is somewhere in here. How come it's not in here? Okay. Relative Okay, so it's like I'm done studying for the night, and now I'm just filling out my active recall questions. And as you can tell, they work because half of these I don't even remember what they were anymore. So that's good. That was what I was looking for. So I have all of these questions, but not enough answers. Has to do with charges in motion, their properties, of the concentration gradient. Deals with equilibrium is such an interesting concept. Can anyone find an example of something in nature that doesn't inherently seek equilibrium, that doesn't seek balance. Like, this has to do with symmetry. This has to do with how things grow and, you know, like offset imbalances. Like, in the brain, there's a such thing called concentration gradient, where certain ions, you know, are not even, but their goal is to reach that equilibrium, that balance. And that's what triggers the process, because they're trying to reach equilibrium. Like, that's crazy and beautiful at the same time. It deals with the electrical charge. That was it for neurons and resting action potential. It's gonna be 10. I think I'll stay live for another 15 minutes, and then that'll be an even 4 hours of content. And then, yeah, I will finish in 15 minutes. Okay, so this neurons and action potential. Oh, so what I did is I set up the questions, but I didn't fill in the answers. That's what I did. Okay. What are the primary ions involved in the resting potential of neurons? At rest, the neuron's membranes sodium channels are closed and therefore mostly impermeable to sodium. But the sodium potassium pump continues to pump in K, so there's a higher concentration inside the membrane. Okay. You know what just popped in my head? I want to watch Van Helsing. For some reason, I want to watch Van Helsing, or I want to watch Underworld. I don't know why. That just popped in my head. Okay. What properties determine which ions are inside and outside the membrane? By the electrical gradient, both sodium and potassium are cooled inside the cell. But sodium channels are closed and at rest, while the sodium potassium pump still brings potassium in. So by the concentration gradient, potassium is more likely to be pushed out because of its high concentration inside. The potassium inside and outside the cell almost reach a balance because the concentration gradient pushes it out, but the sodium pump continually brings in two potassiums. What properties do we check? So, simple answer, the electrical gradient. Perfect. And why? Okay. What role does the concentration gradient play in the resting potential? Potassium tends to flow into the cell because of the electrical gradient. Protein and chloride ions are negative and therefore attract a positive charge, but tends to flow out because of the concentration gradient. Yes. I think I'm going to combine these. I'm actually going to move these up here. Turn these into polarized. Okay. What role do the electrical concentration gradients play in the potential? Okay, tends to flow because of the electrical gradient. It tends to flow out because of the concentration gradient. How does the sodium potassium pump function? The difference is set up by the sodium. It pumps 3 sodium out and 2 K in for each activation. The selective permeability of the membrane prevents sodium ions from remaining pumped back right in. Once pumped out, they stay out. When the neuron is excited, trigger sodium channels open. I can't waste them. Okay, I can do this. How does numbering reach 0 net force? Because tomorrow I have to wake up at 9:30. It's whatever. Ooh, is anyone saying anything? be saying goodbye and good night, and thank you for joining. As always, like, subscribe. I am very tired. I should talk to the camera. I keep forgetting that the camera exists. Yeah, thank you for joining. I hope you enjoyed the stream. I hope you enjoyed the music and the quality. I hope it was great. You're welcome to join me next time. Follow, subscribe for updates. For when I subscribe. When I stream, I'm brain dead, as you can tell. I stream primarily Mondays through Wednesdays after 5 p.m. because I usually have classes in the morning, and then for the rest of the week, my schedule varies with my work schedule. So sometimes I stream in the morning, sometimes I stream in the evening. But yeah, that's what I do. So yeah. Anyway, have a wonderful night. Bye.